A single shot that keeps working for 3 months sounds almost too good to be true. For a patient, it's freedom from a daily pill. For the scientist who has to prove exactly how that drugs come out, drop by drop, over 90 days. It is one of the hardest measurement problems in our field. Hello, I'm Raghuveer Singh Parmar, Director Scientific Affairs at ResolveMass Laboratories, and we work with pharma and biotech teams every day to characterize long-acting injectables and prove how they perform. Today in this video, which is one of our long-acting injectable series, we'll see how scientists actually measure drug release and performance. One injection and months of release. So what makes these products so different? A long-acting injectable is not a tablet that dissolves once. It's a depot, a tiny reservoir of drug built from polymer microparticles, lipids, or in situ forming gel that sits in the tissue and releases medicine over weeks or months. And here is the catch. That depot is alive in a sense. It swells, degrades, and changes every single day. A product you test on day 1 is not the same product on day 60, which is exactly why ordinary dissolution methods fall apart here. 3 questions. To characterize something that keeps changing, you cannot lean on one measurement. You have to answer 3 questions at once. Which is actually in the depot? The chemistry, how big and how uniform are the particles? The physics, how fast does the drug come out? And the release. Those map to 3 analytical pillars: LC-MS, particle size, and morphology. And in vitro release testing. Get all 3 talking to each other and you understand the product. Miss one and you have a blind spot. So pillar 1, LC-MS. Let's start with the chemistry. Inside a depot, your drug is surrounded by a polymer, lipid, and surfactant. And in a simple UV detector, all that noise can hide the very thing that you are measuring. This is where LC-MS earns its place. It separates compounds by mass, so the drug signal stands out cleanly even in a crowded matrix. A triple quad MRM mode identifies and quantifies trace drug in the release sample. High resolution MS catches the polymer drug degradants that form quietly during the storage. The real trick is dissolving that depot without fouling the instrument. Pillar 2 is size, shape, and surface. Next, the physics. Particle size is the critical quality attribute because it drives almost everything that matters. How easily the product injects, how uniformly it is loaded, and how fast it releases. But no single sizing method tells the whole story, so we work orthogonally: laser diffraction, dynamic light scattering, and electron microscopy, each catching what the others miss. Zeta potential warns us when particles will clump on the shelf, and for intramuscular and subcutaneous microspheres, the sweet spot sits between 10 and 250 microns, small enough to inject, large enough to form a depot. Pillar 3 is measuring drug release. Now the heart of it, measuring drug release, there is no compendial recipe here, and biggest challenge is maintaining sink conditions. Keeping fresh medium around the depot for months without changing how it behaves. We reach for USB4 flow through cells, dialysis setups, or sample and separate methods, often in biorelevant media spiked with albumin and enzymes to mimic the injection site. And we sample hard and early 1, 2, 4, 8, 24 hours to catch the burst, because release comes in three acts: a burst off the surface, a long diffusion phase, and a final erosion as the polymer breaks down. A good method captures all three. Different matrices, different traps, and no two formulations behave alike. A PLGA microsphere gives that classic three-phase curves. And high-resolution MS often reveals drug bound to lactic and glycolic acid fragments, and in situ gel traps part of the dose and dissolves the rest. So you must separate the two before any number means anything. A peptide or GLP-1 payload needs ion pairing LC-MS just to confirm it is intact. At ResolveMass, we have watched a method that fits one matrix quietly mislead on another, which is why we match the analytics to the formulation, never the reverse. From data to submission. So how do you turn all this into something a regulator accepts? You build it as a system using quality by design. Start from the target product profile, the release duration you are aiming for, define the critical quality attributes, particle size, drug loading, release profile, link them to the process parameters that control them like homogenization, speed, and polymer ratio. Then connect your in vitro data to in vivo performance through IV/IVC until your release test genuinely predicts the clinic. That is the difference between testing the product and controlling it. So what should you take away from this discussion? One injection means months of release you have to measure. No single method to enough for chemistry, particle size, and release have to work together. In vitro release is your performance fingerprint, and teams that build IV/IVC and QBD in early are the ones whose submissions survive the first hard question. If you are developing a long-acting injectable and want a CR or CDMO partner who treats release characterization as real science, not a box to tick, ResolveMass Laboratories supports small and mid-sized pharma teams from first feasibility study through ANDA, MAA submission. Subscribe, and thank you so much for watching this video until now. Goodbye for now. See you in the next one. Until then, goodbye.